know the function, the direct transmission of the sequence, alright? So this is some of the functions where you can use what bioinformatics are utilized, alright? So the field of science, which biology, computer science and information technology merge into a single discipline. So where, okay, for bioinformatics, you need at least two expertise. You need a computational expertise and also biology expertise. If a computation do their work alone, they cannot know what kind of data we want to interpret. Even same as biologists, if we have the data, if we cannot show it, there's no use. Alright, so this is where two fields are combined together, where you get a bioinformatician that study biological questions by analyzing molecular data. Alright, so the entire complement of genetic material carried by a liver kita panggil genome. Alright, so genome is actually the whole DNA, RNA, or cDNA, whatever genetic hereditary component that inside a organism, be it in human or any higher organism or smaller organism. If it consists inside that organism, so it's a genome of that organism. If human lah. If life, flies lah. If it's worms, it's worms. Alright, it's worm genomes. Alright, so it's different between one genome to another genome. Alright, so inside the genome, in eukaryote, you will have up to 3 subcellular genome. Kita panggil nuclear and also mitochondrial and also plastic genome. This is in higher organisms. So each genome encode for their own DNA sequences. Alright, and then bacteria have either circular or linear genome and may also carry plasmid. Like I've been telling you before, ke kelas lain kami bagi tahu pasal plasmid. It can be a circular plasmid inside another. So extra DNA materials inside a bacteria. I did tell here, right? Tak ingat. I think I tell in this class, right? Tak ingat. Did I tell? Oh, okay. Kawan depan signal dia. Okay? Alright, so basically the hereditary genes of all the hereditary materials inside one's organism, kita panggil genomes lah. Alright? Okay. And then of course, when you want to do gene modification or you want to do molecular analysis of a certain organism, you need to understand the central dogma. What is central dogma? Bernafas pun dengar. What is central dogma? DNA makes RNA, RNA makes protein. So basically it's a process from DNA to protein that involves two main process. Kita panggil apa? Translation and kira tu betul. Saya tak tahu you dengar ke tak? I assume you guys tahu lah. Alright. So this is the process. You can see there's the transcription and also a splice. This is for higher organism lah, eukaryotes, alright? And then you have a translation. So these two processes are very important in central dogma. For eukaryote lebih mudah, tu sebab I suka study bacteria. So easy. No lah, it's not just because it's easy. I mean, you need to have passion towards something, right? Okay, since I was exposed to microorganism rather than higher organism, and that's why I prefer microorganism. Alright? So you can see it here, the process. Very straightforward, a bit different here and there. Alright? So and then you need to know about amino acids. So before the molecular editing whatsoever, these things you need to be good at this fundamental thing. Alright? Amino acid, what type of acid. So all the polarity is also important. That is not saja. In the first year we teach you. We do not teach you why do you know base amino acid, why do you need to know positive polarity charge, something like that, amino acid. So all of this is function in genetic editing. Alright? Okay, so these are the things that you need to know. Maksudnya if you need to know, in order for you to explore editing, you kena ada strong background. Kalau you tak ada strong background, you don't have this kind of strong background, it's going to be very hard on you in future if you want to go to this field. right? So if you like to do cloning or genetic modification, CRISPR, so please read a lot about this kind of fundamental knowledge on the central dogma. Alright? Okay, so this is just another example of the amino acid. So these are the things yang you patut ingat. Apa start codon? Okay, stop. Saya selalu ingat start codon. Wak uga. Okay, I don't know, have some pattern that you can always remember. So whenever you have that kind of setting in your mind, it'll be easier for you in future. Alright? Okay, so this is an example of protein sequences. Okay, you can see that reading direction from sequence. So these are the things that you need to explore once you want to study the manipulation study for the hereditary disease. And this is another examples of how do you want to study the changes. Let's say you have the sequences like here. You have these sequences. No use if you don't know how does the functions of the protein. Okay, protein function can be obtained from its structure, from its pattern of shape, the shape of its fold. Right? The functions of protein are one of the factors that involve the reactivity. In order for you to know the function, is based on the folding of the protein. So if you only have this data, it's going to be very hard for you to visually recall that in your memory. Sebab dia ada leucine, cytosine, sepatutnya dia lipat macam tu. So it's going to be very hard for you to do it in your mind. Right? So that's why bioinformatics come handy, where it can help you to visualize into 3D structure like this. Alright? So they will create a database that know the interaction. If let's say leucine, cytosine, this is the folding. Cytosine, serine, this is the folding. So they will fold for you and you will just study the interaction. Alright, it'll be easier. So this is the function of bioinformatics, where you can illustrate your data and you can analyze it. There's no use you have a data but you cannot analyze it. Alright, so that's happened 10 years back before bioinformatics become a huge thing. Alright, people have a lot of data but we cannot correlate it. Why? Because it were not shown. Alright, same. That's why it's very important for us or for researchers to show our data in a way where people can read it, not in a way that that's why you need to visualize it in graph, in figure, in table, so that people can understand your work, not just written in your logbook. Alright, so that's the main concept of why you are publishing your work. Alright, so not just because kalau publish you famous, tak famous pun. Nobody will know about you if you publish a book, but unless you publish in Nature, then somebody will know you lah. Alright, but if let's say you just you have a new study without publishing, nobody know about your result or your work. So that's why we encourage our students to write on your research. Alright, so that's the importance of writing a research paper. Tertibu, bagi semangat lah. Alright, so how does it look like on a computer monitor? So usually ni adalah your basic data. Let's say when you do your DNA sequencing or when you do modify a certain cDNA library. So when you have the cDNA library, this is what the result come in. So with this kind of result, do you think you can do something on it? I mean, if you just put this inside a disease or inside a journal, so of course people ask, so what does this sequence code for? So you need a tool, you need a database in order for you to interpret this data. Same like here. So you try to find a reading frame. Just now, you tahu ATG and also stop codon, right? A reading frame. So there's two terms for sequences when you say it's a CDF or it's a ORF. So CDF apa? Siapa tahu? What is CDF? What is ORF? Open, okay, good. So open reading frame, tahu CDF? Tak tahu. Tak apa. Tak marah. CDF is coding sequence. So there's a difference between ORF and CDF. If ORF You can just putatively say that it's a protein sequence or a gene sequences because they have open and they have start and stop codon. But of course, are they able to be translated into a protein? Are they have function? So if it have function, we call it a CDS, where it become coding sequence. It have a function kalau you tidur. Okay, good. Alright, so basically it's like that. Alright? So whole genome is going to be greater data. This is just 1/100 of the data, maybe 100, 200. So the data will be much more bigger than this. Alright, so I'd like to introduce you to human genome project. Alright, because your friend dah tertidur, so I give a video so that you can be happy with it. Alright? So let's see how bioinformatics are utilized in human genome project.
Confession.
The huge collection of genes inside each and every one of your cells. You probably also know that we've sequenced the human genome. But what does that actually mean? How do you sequence someone's genome? Let's back up a bit. What is the genome? Well, a genome is all the genes, plus some extra, that make up an organism. Genes are made up of DNA, and DNA is made up of long paired strands of A's, T's, C's, and G's. Your genome is the code that your cells use to know how to behave. Cells interacting together make tissues. Tissues cooperating with each other make organs. Organs cooperating with each other make an organism. You. So, you are who you are in large part because of your genome. The first human genome was sequenced 10 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries, and cost over $3 billion. But someday, very soon, it will be possible to know the sequence of letters that make up your own personal genome, all in a matter of minutes, and for less than the cost of a pretty nice birthday present. How is that possible? Let's take a closer look. Knowing the sequence of the billions of letters that make up your genome is the goal of genome sequencing. A genome is both really, really big and very, very small. The individual letters of DNA, the A's, T's, G's, and C's, are only 8 or 10 atoms wide, and they're all packed together into a clump, like a ball of yarn. So, to get all that information out of that tiny space, scientists first have to break the long string of DNA down into smaller pieces. Each of these pieces is then separated in space and sequenced individually. But how? It's helpful to remember that DNA binds to other DNA if the sequences are the exact opposite of each other. A's bind to T's, and T's bind to A's. G's bind to C's, and C's to G's. If the A, T, G, C sequence of 2 pieces of DNA are exact opposites, they stick together. Because the genome pieces are so very small, we need some way to increase the signal we can detect from each of the individual letters. In the most common method, scientists use enzymes to make thousands of copies of each genome piece. So we now have thousands of replicas of each of the genome pieces, all with the same sequence of A's, T's, G's, and C's. But we To do this, we need to make a batch of special letters, each with a distinct color. A mixture of these special colored letters and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome. This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book written in a language you don't speak. You can recognize all the letters, but still have no idea what's going on. So the next step is to decipher what the sequence means, how your genome and my genome are different. Interpreting the genes of the genome is the part scientists are still working on. While not every difference is consequential, the sum of these differences is responsible for differences in how we look, what we like, how we act, and even how likely we are to get sick or respond to specific medicines. Better understanding of how disparities between our genomes account for these differences is sure to change the way we think, not only about how doctors treat their patients, but also how we treat each other.
Interesting, right? So basically, it's very interesting. So if you were to sequence the sequence and analyze it by yourself, it's going to be very. Jadi bukan keluar lagu Korea kan, itu example. Alright, so basically that's it. So how big are whole genome? So you can see from the video just now how big is human sequences. And of course, when the higher organisms are playing around, we also have repetitive genes that don't have function. So usually when you compare our sizes like E. coli, you have around 4.6 to 10 power 6 nucleotide, you have yeast. So the higher the organism, the higher the genes number. But of course, there's, the higher the organism, there's also would be the repeated sequence. So not all sequences have functions, alright? So as a conclusion, what can we actually do for bioinformatics? So these are all the things that you can do with bioinformatics. You can assemble the sequence from the NGS. So the NGS is the sequencer that you seen just now in the whole genome sequence for human project just now. You see the big setup where they use a fluorescent signal to sequence, to detect the ATCG, right? So those are a sequencer, or we call it an NGS, next generation sequencing. And of course, there are so many types of NGS, alright? So basically for NGS, you have PacBio, Oxford Nanopore, and also you have Illumina, MySeq, HiSeq, NovaSeq, whatever seq it is, alright? So it's always developing. So you will get higher, more and more special gene sequence NGS machine where you can get a very confidence data for the ATCG. Okay, so basically you have all of these functions, so you can just read it out. So in next lectures, I'm going to introduce some applications of the bioinformatics. Like what are the databases that we can use for 3D structure, for 1D structure, all those things in next lectures, alright? So here are attendance for today. I see you.